Hello everyone, welcome to my first video on this YouTube channel. Um, this video is going to be about biological molecules and I'm going to be explaining this topic, monomers slash polymers and carbohydrates. So this is one section of the entire topic and I'm going to be uploading multiple videos covering each topic of the specification in kind of small digestible chunks for AQA A-level biology. Now, for most of you, you've just started year 12, you've just probably began a few lessons at your sixth form or college, and this section hopefully will deepen your understanding of this first part of topic one. And yeah, let's get right into it. Now, the first thing that we want to understand is what are monomers and polymers? Now, monomers are smaller units from which larger molecules are made. And polymers are a molecule which is made from a large number of monomers joined together into a chain or in a chain. And now I've included this kind of like rough diagram to kind of showcase these kind of like easy to understand definitions in a very easy digestible way. You may have encountered these definitions in chemistry, whether it's A level or GCSE or even biology, GCSE and A level. So yeah, the red squares that I've just drawn in this diagram are monomers, and the red lines kind of connecting each monomer is a chemical bond. Now, the only thing that you may be kind of um, unsure about is what is this black box around this whole structure? Well, the black box is just like a theoretical kind of way of me kind of highlighting what the structure is. Because this entire thing that I'm highlighting right now is the polymer. Now, this is what I try to do with the with the marker. I try to highlight this area and present it as the polymer. So this whole thing is the polymer. Now, moving on, another part that is very key to understanding is the condensation reaction. Now, the condensation reaction is a reaction that involves the elimination of a water molecule which joins two molecules together via the formation of a chemical bond. Now, what do I mean by this definition? What is the main part? What is the key part of this definition? Well, if you think about it, we're going from separate monomers to a large structure, which is called the polymer. Now, if you've done GCSE chemistry and A level chemistry, you know you're familiar with um, kind of balancing equations and chemical equations. Well, this is what the diagram is showing you. We have four monomers, and we have an arrow um, going towards producing the product, which is the polymer. Now, the polymer is the same as I've presented here, just less monomers that create the polymer. The highlighted in yellow part is just to highlight the chemical bonds, which are just here. And the reason we have free water molecules as a byproduct is because of the elimination of water that involves the formation of a chemical bond. Now, you may say, well, fair enough, we've joined these monomers to form the polymer, but why do we have free water molecules formed? Well, if you think about it, how many bonds have we actually created between these four individual monomers? Well, we've created one, we've created a second one, and we've also created another third one. Therefore, each water molecule is released, or in other words, eliminated, for each of the formation of these chemical bonds. So one is released for this one is released for this one, and another is released for this one. Therefore, producing a net total of three water molecules as a byproduct. Now, you may be asking, what is a byproduct? It's just another product within a chemical equation. Um, and the main product is the polymer. That's why this is the byproduct. Um, that's pretty much it. You can also note these diagrams down, pause the video and note them down. and any information that I'm kind of like explaining throughout might be beneficial to note down as well. Moving on, there's another definition that you need to be aware of. Now, I understand that in this topic there's a lot of definitions, but this is the first topic of the A level kind of specification. So there will be a lot of definitions that you need to remember. And these definitions are kind of key in the understanding of the next topics that are going to come up. So I think taking this section or this kind of topic step by step and in easy kind of chunks is the best thing to do. So this definition is hydrolysis, or let's just say the hydrolysis um, let's just say the hydrolysis reaction, which would be the better way, better definition. So hydrolysis reaction is the breaking of a chemical bond between two molecules involving the addition of a water molecule. Now, reading that at first, kind of as a first impression, you may find that this reaction is the opposite of what we just learned, which is the condensation reaction. So therefore, this information is kind of like the definition is the opposite of each other. So roughly, you can kind of understand the diagram is quite similar. It's just going the other way. So we have three water molecules to break three individual um, kind of chemical bonds within a polymer to produce individual monomers that are separate and that are not joined together. So therefore, the reason we add the water molecule is to break it, and when we make a chemical bond, we release or therefore eliminate a water molecule. So in this case, we're forming them, therefore we add a water molecule. So we formed one, two, three. We add three water molecules to the polymer, and then we produce a product of four individual monomers. And you can also note this down. Now, moving on to carbohydrates. So, kind of a few definitions, extra definitions that you should be familiar with is an example of a polymer. Now, an example of a polymer is carbohydrates. Now, carbohydrates, you may have noticed, um, are kind of like mentioned on nutrition food labels or drink food labels, complex sugars essentially, and they can be referred to as polysaccharides in AQA level biology. Now, you may think, what is a carbohydrate made up of? Well, since it's a polymer and a polymer is made up of monomers, we can see that kind of monomers make the polymer, therefore monosaccharides make polysaccharides. And I hope that you can follow that kind of explanation easily, because since we have a monomer and we form polymers from monomers, therefore a polysaccharide, which is a polymer, is formed by many monomers. So therefore, this is the definition of a polysaccharide is a polymer formed by many monosaccharides joined by a glycosidic bond in a condensation reaction to form chains. Now, you may be thinking, okay, monosaccharides are monomers, and these monomers form polysaccharides, but what is a glycosidic bond? Well, a glycosidic bond we kind of briefly mentioned, but didn't specifically kind of mention that it's a glycosidic bond. So it's these chemical bonds that are highlighted in yellow throughout these kind of previous diagrams. This is glycosidic bonds are specifically only in carbohydrates. So therefore, glycosidic bonds will only be seen in carbohydrates in AQA level biology between monosaccharides of polysaccharides. Uh, Therefore, you also may be asking, what is a disaccharide? Well, a disaccharide is a sugar formed from two monosaccharides joined together by a glycosidic bond in a condensation reaction. So it's essentially just two monosaccharides joined together, whilst a polysaccharide is many monosaccharides joined together by a glycosidic bond, which has been formed from a condensation reaction. And polysaccharides form chains. So that's the reason the chain bit is also included within the definition. And moving on, we have examples. Now you may be like, whoa, what is this? Don't worry, don't kind of You don't have to go all over the place with this information. Just digest it one at a time. The first bit, we have monosaccharides examples. Now, these are the only examples that you need to know for AQA A-level biology. It's alpha glucose, beta glucose, fructose, and galactose. Now, you may be like, what is A, what is B? Well, A is for alpha, B is for beta. So that's why we have alpha glucose and beta glucose, fructose and galactose. And then your next question might be, what is alpha glucose? What is beta glucose? What are the differences between them? So alpha glucose and beta glucose, fructose and galactose are monosaccharides. And the difference between glucose, which is alpha and beta, is down below. So here I've included a kind of uh, diagram that you need to know how to draw um, in the exam. Now, alpha glucose is on the left and beta glucose is on the right. And the main difference between the two is that the OH group is inverted on carbon 1 on beta glucose. Now, you may be kind of familiar with these structures if you do A level chemistry or GCSE chemistry. This is like the skeletal formula of A level chemistry. You'll kind of encounter this formula later on. At first, if this is your first lesson of kind of biology, you might be frightened of these diagrams, but don't be. This is the only thing that you'll be expected to draw. And it also comes up not really often, so but it's very highly kind of like recommended to know because it's quite easy. So you just draw these as they're shown, and the only difference that we can see, which is clearly if I switch color to yellow, is this bit. So we have OH down here and OH above. Now you may be like, gosh, they look so similar. How am I gonna kind of memorize these structures or be able to differentiate between them? So using the alphabet, alpha, beta, I use it as well to kind of differentiate, and I have a trick. So going from bottom to top of this structure, this is A, and this is B. This is A, and this is B. And if it's alpha glucose, I know that OH is at the bottom because this is A. And if it's B, I know that the OH group on beta glucose is on the top because it's denoted by the letter B that is representing the top half of the molecule. You can also, some students also like to divide it into kind of like two different sections, but I feel like that kind of overcomplicates it. I think just the A and B, kind of like bottom half, top half. So the top half is for B, the bottom half is for A, is much simpler. But that's just like a personal preference of mine. You can do either, or even kind of memorize both tricks to remember. And you may be thinking, what is carbon 1? Well, it's just the first carbon within this structure. Um, and that's pretty much it. I wouldn't worry about kind of understanding carbons just yet. I feel like if you do chemistry, your chemistry teacher will explain what carbon 1 is and carbon 2 and how to kind of understand chemical structures or displayed formulas. But for biology, you won't go in depth in terms of um, the chemistry stuff, kind of like directly. So you just need to kind of understand the difference between this. Now, you may be thinking, what is this definition of isomers? Well, the isomers bit is related to both of these. So an isomer is a molecule with the same molecular formula but differently arranged atoms. And you may be thinking, what is a molecular formula? I've never heard of that before. That's completely fine. It's quite a simple definition. A molecular formula essentially shows the exact number and type of atom present in a molecule. So, if you've done GCSE chemistry or A level chemistry, you will come across molecular formulas and also different type of chemical formulas. But let's just focus on molecular formulas. So they can like you may come across them in chemistry. There is like C8, H8, or C8, H16. C and H is the individual kind of elements, and 8 is the number of each atom present of the element in the molecule. That's pretty much it. If you're just solely doing biology, I wouldn't worry about this too much. Just memorize the definition and kind of understand the kind of broken down, simplified definition that is just needed for you to understand, because it's not really usually asked in biology. You just need the understanding of what it is, essentially. But this part, the second bit, which talks about differently arranged atoms, you can kind of visualize, because O and H is an atom, H is an atom as well. So at the bottom for A, we have O and H, but for beta glucose, we have it at the top. So therefore, the atoms are arranged differently within the molecule, despite having the same number of atoms for each element as in alpha glucose and beta glucose. That's pretty much it. Not too complex, but it should take some time to get used to if you're new to biology. This is another chemical equation, but just kind of presented in a much more complex and detailed way than the simplified diagrams at the beginning. So here we can see that we have one alpha glucose reacting with another alpha glucose to produce one molecule of maltose and water. Now you may be like, what is maltose? Well, maltose is a disaccharide, as we've talked before. And disaccharides are formed from monosaccharides. And here we can see that two molecules of alpha glucose form to form the maltose. Now, you also see that water has been released. And if you've analyzed the equation, you might suspect that this is a condensation reaction, and you are in fact correct. This is because this is a condensation reaction because a chemical bond is formed whilst releasing one molecule of water per each chemical bond formed to form a disaccharide or a polymer. So in this case, it's a disaccharide, as I've mentioned, and you may be like, okay, where is that chemical bond? Previously, I've highlighted them in a kind of in a straight and simplified way. In this case, we see that the diagrams are much more kind of complex, and you may see something like this in your exam. Don't be terrified though; they're quite kind of easy marks, in my opinion, and in a lot of students' opinion, and they're kind of easy to quickly do. They're time efficient. So the only difference that you see between these two and this is that everything is the same except this kind of bit that I'm circling in red and this bit that I'm circling in red. This is the only difference. You can analyze it once more to kind of see if I've missed something out, but there isn't. And you can clearly see that this is the only difference. And think about what kind of happened to kind of go from the left hand side to the right hand side. Well, you can clearly see that we've removed one molecule of water, and this kind of looks like this in chemistry. This is the displayed formula. And this is representing oxygen, O, and this is representing hydrogen, therefore water, H2O. Now, the only thing that we're left with here is O. So therefore, what have we removed from this section? If I quickly remove the red bits, I'm going to highlight what has been removed. It's this H, including this OH. Now, if this is removed, you can have a look at what is left. The only thing that is left is this O. And this O is literally here. That's pretty much it. And this highlighted bit forms this H2O. That's pretty much it. And the rest is the same. Literally. Everything else is the same. And you just redraw the whole molecule perfectly identical to these. And they just join together at this bit. That's pretty much it. And that is the only thing that you'll be expected to kind of understand for these diagrams in exams. Now, moving on to the symbol that may kind of that you may kind of misunderstood or weren't familiar with. So, moving on to diagram symbols. Now, in this previous kind of equation, right about here, we've seen it here, and I'm gonna explain it right now. So it's a reversible reaction, and a reversible reaction is a reaction in which the reactants can form the products, and the products can form the reactants. Now, what do I mean by this? So this side is the reactants, and this side is the products of the equation. And think of this as like a kind of like an equal sign in a sense. This middle bit, and the left hand side is the reactants, and the right hand side is the products. So therefore, it's a reaction in which the reactants can form the products, and the products can form the reactants. And since I've kind of mentioned already that the forward reaction is the condensation reaction, I'm just going to represent it in a short form. Therefore, if the products can form the reactants, what would the opposite reaction be? Well, the opposite reaction would be the opposite of condensation, because we're doing the opposite of the condensation reaction, which is effectively hydrolysis. Hydrolysis. And that's pretty much it. And you might be kind of like confused. Why is it hydrolysis? Well, we are going from a disaccharide, or essentially a larger molecule, to much smaller individual molecules via the breaking of a chemical bond. And this chemical bond is the glycosidic bond, which is this bit that I'm showing you right now. This is the glycosidic bond. And as you know, hydrolysis involves the addition of a water molecule. And as you can see, that the water molecule is present for the addition. So therefore, the forward reaction, which is that way, from left to right, is condensation, and the backwards is hydrolysis. And you might be stumbled or kind of confused about the glycosidic bond. It's pretty much just this bit. Nothing to worry about or kind of be confused about. You don't need to know kind of the in-depth definition or anything else. Now, this is kind of a short kind of summary of what I've just kind of explained. You can note this down. The top bit of that symbol represents the forward reaction, which in this case was the condensation reaction of disaccharide from two monosaccharides. So therefore, two monosaccharides form the disaccharide. Now, this bit is the backward reaction, and in this case represents the hydrolysis of a disaccharide to two monosaccharides. And that's pretty much it. Very simple. Now, this bit might confuse you a little bit, but it takes a little bit of time to kind of get used to and kind of memorize. We have monosaccharides, and as we know, disaccharides are formed from the joinage or kind of joining of monosaccharides via a chemical bond. Now, we see that we have two alpha glucose reacting to form maltose and water. Now, this is essentially what we've just shown here. It's just a word equation, not the symbols and the actual kind of complex diagrams. So this is just something you need to memorize. And the type of disaccharide formed is a reducing sugar for maltose. Alpha glucose plus fructose gives you sucrose and water, and it's a non-reducing sugar. Alpha glucose plus galactose gives you lactose plus H2O, and which is a reducing sugar. Now, reducing sugars are sugars that donate electrons, and the reverse is true for non-reducing sugars, which lose kind of electrons. And if you think about it, the type of disaccharide form is kind of easy to memorize if you kind of color code it. Now, I color code reducing sugars for orange and non-reducing sugars for red. And I know that the first disaccharide form and the last disaccharide form are reducing sugars, whilst the middle is non-reducing sugars. That's pretty much it. and that's how I kind of remember it. And you just kind of need to memorize this bit. I know it's annoying, but trust me, once you memorize it, you're good to go. And it shouldn't take much time. Now, you may be kind of confused. What are monosaccharides? What are disaccharides? And you kind of want to get an idea of their sizes. Now, going from smallest to largest in size, monosaccharides are the monomers, therefore the monomers are the smallest. Disaccharides are two monomers, so therefore two monosaccharides joined together by a glycosidic bond. Therefore, they're the kind of mid-size, they're in the middle. Whilst the polysaccharides are the large polymers. And large polymers are many monosaccharides kind of joined together via glycosidic bonds. And they're usually in chains, therefore indicating that there's more than one, or sometimes even just one. But these are far greater in size compared to monosaccharides and disaccharides. And this is kind of simple to understand. Whilst I kind of draw kind of squares for you in a sense. So if I was to represent monosaccharides by a square, this is how it would look like. Disaccharides by two squares, as it's two monosaccharides joined together. And polysaccharides are just many multiple monosaccharides joined together. Therefore, it will just be many. And this can go on to thousands or possibly hundreds and thousands of monomers joined together. And this would carry on. So therefore, you can clearly see the size increase. And it's something that is very easily kind of visualized in a sense. Now, this bit also includes memorization, of course, which is polysaccharide examples. Now, we have three examples of polysaccharides that you should be aware of. The first is starch, the second is glycogen, and the third is cellulose. Now, everything that I'm showing on the screen is something that you need to memorize and kind of be able to answer, and that could come up in the exam. So none of this is irrelevant or kind of extra information. So starch is a polymer of alpha glucose. Therefore, it contains many monomers of alpha glucose. And the function of starch is it acts as an energy store in plants' cells. Now, what is the structure of starch? Well, the structure of starch is a mixture of amylose and amylopectin. Now, you might be asking, what is amylose? What is amylopectin? Well, amylose and amylopectin are just two different chains, which are kind of within the structure of starch. Now, the first amylose is unbranched, helical, and has 1,4 glycosidic bonds, whilst amylopectin is highly branched with 1,4 and 1,6 glycosidic bonds. Now, you might be thinking, what is branched? What is unbranched? What is helical? What is glycosidic bonds 1,4 and 1,6? Now, I'm here to explain that. So, unbranched is presented via amylose. You can just see it's got a quite linear kind of forward structure. There's no kind of other bits hanging out. While for amylopectin, you see this. I'm gonna highlight it. It's quite visible, so it's quite visible for you. Is a straight structure similar to amylose that is straight right here, but we have these extra kind of strands that interconnect. Now, these interconnections of is basically the branching, and this branching is very important, and it contributes to its structure and to its function essentially. Now, this is the reason why we call amylose unbranched, because it doesn't have these red kind of connections to the main kind of linear chain, whilst amylopectin does. Now, helical, it just refers to its kind of helical structure of amylose. That is something you can search up and see many images of, but it's quite complex. I think it's just easier to remember that amylose is helical. Um, it's just a kind of a term that you need to be that kind of like remember for amylose. And not only that, I think it might be quite beneficial to see the picture, but you shouldn't kind of learn how to draw it or anything. That's kind of out of your scope for A level biology, especially for AQA. And you may be thinking, what is 1,4, 1,6 glycosidic bonds? Well, it's right here. 1,4 and 1,6 refers to what? Is literally the numbers referring to which carbons on glucose the glycosidic bond forms between. For instance, 1,4 glycosidic bonds occurs between carbon 1 and carbon 4 on glucose. So the glycosidic bond is formed between carbon 1 and carbon 4. That's pretty much it. Now, how's this structure related to its function? And these are kind of exam points that you could see coming up in mark schemes. So this is something that you should memorize, definitely. So helical structure means it's compact for a storage in cell. And this is very beneficial because it's very compact and easy to store. The fact that it's a large polysaccharide or large polymer means they can't leave the cell, and it's also insoluble in water. Therefore, the water potential of the cell is unaffected. So these are kind of like the advantages of the structure and of the polymer. Now, moving on to the second polysaccharide that you should be aware of, it's glycogen. And glycogen you might have encountered in GCSE. Glycogen is a polymer of alpha glucose as well, and the function is that it acts as an energy store in animal cells. So the structure of glycogen is that it's highly branched, 1,4 and 1,6 glycosidic bonds, and that's pretty much it. That's the structure, and it's very similar to amylopectin. Like if you want a visual kind of reference, a rough visual reference, and these are the points required to relate it to its structure. The fact that it's branched, it means that the ends are rapidly hydrolyzed to release glucose for respiration to provide energy. And since it's large, it means that it can't leave the cell and insoluble, therefore water potential of the cell is unaffected. Now you may be kind of thinking, what is water potential? You kind of understand water potential later on within this course. And you shouldn't kind of go in depth about it right now or kind of watch the video right now. I think you should just kind of go step by step. And it's something you just need to memorize for now, pretty much. Now, cellulose is also the third type, and it's a polymer of beta glucose. Now, the previous were a polymer of alpha glucose. You must remember that this is different for cellulose. Cellulose is the polymer of beta glucose, which is the different type, essentially, the isomer. So the function of cellulose is to provide strength and structural support to the plant cell wall. Now, you might have heard of cellulose at GCSE biology, which was involved in naming kind of the parts of organelles for plants and the uses of like the cell wall and what it contains. Now, the structure of cellulose can be presented through here, through this diagram that I've kind of found online. And it's long, straight, and unbranched chain. It has 1,4 glycosidic bonds only. It has many hydrogen bonds linking parallel strands to form microfibrils. Now, the many hydrogen bonds are not kind of visible, but if I was to kind of roughly kind of just for a rough reference or kind of rough understanding, they would be right here between the chains, connecting kind of each chain. And the reason that they're there is to kind of provide strength to the plant cell wall. And the hydrogen bonds are strong in very high numbers. So that is very key to your understanding. So many hydrogen bonds forming microfibrils, they provide strength to the plant cell wall, which is the second point. And another advantage of this kind of structure is that it can resist osmotic pressure. And since bonds are difficult to break, it can resist digestion, action of microorganisms slash enzymes. Now, you may be thinking, what is osmotic pressure? That you will kind of encounter later on with the same kind of topic that kind of explains what water potential is effectively. And it's just something you should memorize for now, essentially, if you're at kind of a new stage with biology. Now, moving on, I hope you remember the reducing sugars and non-reducing sugars that we met kind of early on in this video. So I'll give a quick summary. So the reducing sugars include all monosaccharides, which is glucose, fructose, and galactose, and only some of the disaccharides, which is maltose and lactose. Now, the non-reducing sugars include one disaccharide that you need to know, which is sucrose. Now, we have two different tests for these. The first test is for reducing sugars. Now, the first part is that we add Benedict's reagent to the sample. It's just something that you need to know that is a reagent. Now, we just add it to a sample within a test tube. We add the test tube into a boiling water bath, and then we wait for a positive result. Now, the positive result is green, yellow, orange, or brick red precipitate. This precipitate kind of varies in color because it depends on how much sugar, reducing sugar specifically, is present within the sample. That is pretty much it. That's how you test for the reducing sugars. This is like kind of what you would write down for the answer, and something that you will find on the mark schemes. Now, for the second bit, which is non-reducing sugars, we perform a separate kind of test for. So we perform the Benedict's test for reducing sugars, let's just say theoretically, and we find a negative result, which is the sample remains blue. Now, the question might ask us, since the sample remains blue, which indicates that there's no kind of reducing sugars present, how would we test for non-reducing sugars? Now, this is where you come in this kind of stage, and you explain the kind of key points. So you would add the 3 drops of hydrochloric acid and specifically dilute. And this is key to mention within your kind of answer. And the reason this is there is to hydrolyze the sample into its constituent reducing sugars. Now, the next step is we would heat the sample in a boiling water bath. And then we would neutralize the excess kind of dilute hydrochloric acid, which is acidic, with sodium hydrogen carbonate. Now, sodium hydrogen carbonate is an alkali, and when an alkali is added to the excess acid, the acid will be neutralized, and we would produce a neutral kind of pH for the sample. Then we would add Benedict reagent, and it says we would heat it again. So we add it first, then we heat it again. And then number 6 kind of shows you the result that we kind of expect. So if it's a non-reducing sugar that is present, we would have green, yellow, orange, or brick precipitate, brick red precipitate, depending on the concentration of the non-reducing sugar present. And so if it was a kind of negative result, the sample would remain blue. That's pretty much it. This is kind of like as in depth you would be required to kind of understand. One more thing is the biochemical test for iodine. Now, this test is just essentially called the iodine test. Now, how do we test starch? Starch, it was the polysaccharide that we saw as an example, and we can test it by this test. Now, how do we do it? We just add iodine to the sample and shake and stir. So the first thing you would do is add it to the sample, then you would shake and stir, and then the third bit would be mentioning your positive result. The positive result would be a blue-black color, and that is pretty much it. And this is like the test tube, this bit, which is like the container, basically, the test tube, whilst the blue bit is kind of presented as the color change, this bit right here. And that is pretty much it. That would be the positive result if starch was present. So, thank you for watching the video. And the next video will be a continuation of this topic, which is lipids. And if you have enjoyed this video, you can kind of comment down below and kind of give me feedback on how I can improve the video or kind of adhere to your teaching style. Adhere my teaching style towards your needs, such as making it more visual or whatever. Any suggestions that you have, I'll try kind of take it into account and see what I can do. So the reason I started this channel is because I want to support Gaza and Ukraine, and I want to support the people in need of kind of urgent help. So you can click the link down below in the description or the comment section, and the kind of link will guide you towards a trusted source, which is the British Red Cross. You should be familiar with this if you're living in the UK, and all of the money that you'll donate will go towards the support of the people in need. Pretty much, uh, that is the video, and I hope you kind of enjoyed it.